Good morning and welcome to the Animal Health course. This is a 30-credit course consisting of parts of immunology, parasitology and biosecurity. Because it's a 30-credit course, you'll get twice as much information, and if you want to do well in the course, you will need to work twice as hard. Okay? Immunology, parasitology, biosecurity are all very important subjects, but they're all fairly demanding. And in particular, immunology and parasitology have their own literature, their own jargon, their own words, and you'll have to work quite hard to come to grips with this. Having said that, they are fascinating subjects. They are important subjects. They are important for animal health, but for many of you, understanding how your own bodies work will be part of the attraction of the course, and in particular, understanding immunology will help you to understand why certain people, certain animals get disease, and perhaps more importantly, why some individuals don't get disease. Before I start on the course, I would like to apologize for the bit of the muddle with the timetable and the website. Part of the problem is now, under the revised university system, lots of people are contributing and they're not all pulling in the same direction. However, we have all those problems sorted out. The timetable has been posted and the website will get steadily clearer as you become more used to it. The first part of the animal health course is a series of 16 lectures in immunology. These are going to follow the textbook, Veterinary Immunology Principles and Practices, second edition by Day and Schultz. They're going to follow it quite closely. Because of COVID, because of changed university regulations, we have completely revised the course. The lectures have been revised and the practicals have been revised. And the immunology lectures now closely follow the textbook. Because there are only 16 immunology lectures and 22 chapters in the textbook, some lectures consist of 2 chapters. And 2 chapters in the textbook we're not going to cover, one on cancer immunology and one on case studies, which is the final chapter in the book. But the other chapters will be covered in the lectures. So in addition to reading the lecture notes, and you will be examined on what is in the lecture material, you're also strongly advised to read the textbook. Having said all that, good luck. If you have any problems, email me. We will hold a series of tutorials on an ad hoc basis during the term when it looks as if some of you are having problems with some of the material. We're here to help. So I hope you enjoy the course. The first lecture is about innate immunity. Now, the immune system can be divided into 2 parts, the innate immune system and the adaptive immune system. Historically, they've been treated as separate subjects, but more and more we're beginning to realize that the 2 parts of the immune system are intricately linked. So the innate immune system has a big say, not only in defending immediately against infectious agents, but also in determining the type of adaptive immune response that is elaborated in response to infections that the innate immune system cannot fully deal with. The objectives of this lecture, and I will give you the objectives before every immunology lecture, are the same as the intended learning outcomes. And there are 3 objectives for this lecture. I want you to be able to distinguish between innate and adaptive immunity. I'm going to give you examples of innate immune mechanisms, which I expect you to understand, as they will be part of the exam at the end of the course. And I'm going to identify 4 barriers that the innate immune system can be thought of as consisting. Okay. Now, the immunity is immensely important for veterinary science and livestock agriculture, as well as for human medicine. Immunology is a relatively new discipline. It's really only grown up in the last 70 years or so as an outgrowth of microbiology and to a lesser extent of parasitology. Immunity is immensely important. Colostral immunity, for example, calves in particular, but also piglets and lambs, suck colostrum, which is the first milk coming from the mother. Colostrum contains antibodies, and these antibodies are essential for the health of the calf, the piglet, and the lamb. Animals that are unable to suck colostrum have much higher death rates because colostrum contains factors such as antibodies, which we'll talk about in the next lecture, which help prevent infections. Immunity, the immune system, underlies resistance to infection. It's fairly clear to most immunologists that without an immune system, life would become impossible. We would be unable to resist infections. But immunity goes beyond resistance to infections. We can use the immune response to help in the diagnosis of disease, because infection with pathogens, these are organisms that cause disease, organisms that cause pathology, signs of disease, infectious agents generate immune responses. And we can use these immune responses to determine whether or not an animal has or has been exposed to or has had the disease in the past. And of course, immunity underlies vaccination of animals, as well as other control measures, such as selective breeding for disease resistance. Now, this slide here is the first figure in the book, and it's an important figure, and we'll refer to it on several occasions. What Day and Schultz have done is divided the immune system into the innate component and the adaptive component. And the innate system is on the left-hand side, labeled innate, and the adaptive system is on the right-hand side, labeled adaptive. And I'll go through this in some detail. We have an antigen indicated at the top as this red double-spiked molecule. The antigen crosses the epithelial barrier at the body surface and then triggers an immune response. In this case, there are a series of innate components, including complement and antimicrobial peptides, a whole series of proteins and other molecules, some enzymes such as lysozyme, which can digest bacteria and foreign material, and there are cells of the immune system. Here we've labeled neutrophils, macrophages, and NK cells, which are natural killer cells. There are other cells as well, which have not been shown here, such as eosinophils, basophils, which also play a role in the innate immune system. For the adaptive immune system, the antigen is picked up by dendritic cells, and dendritic cells here are at the border between the innate and the adaptive immune system. Most immunologists consider themselves, the dendritic cells, to be part of the innate immune system, but more and more immunologists are beginning to recognize that dendritic cells are essential, as is much of the innate immune system, for the proper functioning of the adaptive immune system. Now, the dendritic cells pick up the antigen and transfer it to the lymph node, shown here as this roughly kidney bean shaped. Now, within the lymph node itself, and we'll talk about the lymph node and the lymph in a later lecture, but within the lymph node, the dendritic cells present the processed antigen to T cells, and T cells provide help to B cells as well as other T cells. And B cells then, with the help of T cells, produce antibodies against these antigens. B cells leave the lymph node, mature into plasma cells, and we have specific antibodies shown as these Y-shaped molecules. We have T and B cells, and T and B cells leave the lymph node, those that have been activated, and go into the blood, shown here as this circular shaped organism with 3 endothelial cells around it. And slightly to the right, possibly hidden by my little picture, which I'll hide, are Tregs, and Tregs are the regulatory T cells. Now, this may seem a little bit overwhelming to you, but what we will do is spend the next 15 lectures, as well as part of this lecture, talking about each of these components in turn, until you have, or until you should have, a very good idea of how the innate and the adaptive immune system work, and in particular, how they help you to resist infections and disease. As the slide shows, there are 2 types of immune responses, and they're characterized by different factors. So the innate immune system has non-specific recognition. So enzymes, for example, will recognize specific components, but they don't discriminate between particular organisms. There's no memory of previous assaults. The innate immune system is fast-acting, virtually instantaneous. It's present at birth, and it's the first line of defense. And the innate immune system used to be disregarded by immunologists as relatively unimportant. But it's now clear that it stops most potential threats. And individuals who have deficiencies in part of the innate immune system often find their health is seriously compromised. Now, we are generalizing a little bit here. You can quibble with each of these descriptions, but in general, they provide a good overview of how the innate immune system works. Now, the adaptive immune response, or the acquired immune response, in contrast to the innate immune system, it has specific recognition. So we elaborate specific molecules as part of the adaptive immune system, which recognize specific foreign molecules in a special way. The acquired immune system, the adaptive immune system, has a memory of the previous responses. And if it's seen a molecule once or more often before, then it mounts a much stronger and qualitatively different type of immune response. So we say that the adaptive immune system has memory. It's slow acting compared to the innate immune system. Often it takes 7 days, for example, before antibodies are produced in sufficient quantity to play a role in preventing infection. But it's much more powerful in attacking the antigen. So if often or occasionally the innate immune system won't be able to prevent a microbe from spreading, but the adaptive immune system will kick in and it will control the spread of the antigen. And mostly it develops over time. Now, for both the innate and the adaptive immune systems, there are two types of immune mechanism. There are soluble factors. These are humoral factors. And in the innate immune system, these would include things like antimicrobial peptides, like cathelicidin, and also enzymes like lysozyme. For the acquired immune system, the classic soluble factor is antibody. But there are also cells, and the immune cells give rise to cell-mediated immunity. Now, for the adaptive immune system, essentially, that's about lymphocytes and subsets of lymphocytes. For the innate immune system, as I mentioned, there are a number of cells that contribute to cell-mediated immunity. There are neutrophils, there are eosinophils, there are basophils, there are macrophages, the granulocytes, the macrophages, the dendritic cells, all form part of the cell-mediated component of the innate immune system. But both the innate and the adaptive immune system respond to foreign substances. It's perhaps an exaggeration to say the innate immune system recognizes them, although this is becoming more and more plausible. But classically, we talk about recognition by the adaptive immune system. In addition to recognizing foreign substances, the immune system attracts foreign immune cells to the site of infection by foreign cells. And once there, the immune cells and the soluble factors attack foreign proteins such as toxins. They attack viruses, bacteria, fungi, protozoa, and parasites. And all being well, they control them and prevent them causing disease. Now, the innate immune system can be thought of as 4 barriers of primary defense. There are the epithelial barriers. The epithelium is the outer layer of cells that protect the body, without which you'd just be in an amorphous blob. So your skin holds your body together. You've got epithelial barriers. But mathematically speaking, or speaking in terms of the topology, you have, for example, your gastrointestinal tract, which in some sense is outside the body. You know, it's a hollow tube running from your mouth to your anus. And this too is lined with epithelium. The lungs are lined with epithelium. So you have epithelial barriers, not just your skin on your external surfaces. In addition to the epithelial barriers, there are physiological barriers. We'll talk about these in a little bit more detail. But for example, the stomach is relatively free of most parasites, most microbes. And in part, this is due to the physiological barrier caused by the low pH, by the acid environment. There are also phagocytic barriers. The phagocytes, such as macrophages, neutrophils, play a major role in clearing foreign infections. And there are also the inflammatory barriers. Inflammation is often seen as a nasty thing, but inflammation is a normal component of the response to infection. So you have your externally exposed skin, for example, but you also have your mucous membranes, your lungs, your gastrointestinal tract. Women, you have your vagina. And these are all, in a sense, external surfaces, external to the body, although we don't normally think of the surface of the lung as being an external surface. In technical terms, it is. So you have the externally exposed areas. Your mouth, for example, the inner part of your mouth is externally exposed, and some people think your mouth is one of the dirtiest places in your body. But these surfaces need to be protected. So the skin has its protection. The skin is often dry. Bacteria don't do very well in dry environments. But the skin often is covered in a layer of fatty acids, the sebum. The mucous membranes are wet. They're covered with a layer of mucus, which can entrap bacteria and to some extent larger parasites, hinder their movement. And the lung, for example, is lined with ciliated epithelial cells. And I've shown two pictures here on the right. One of them at fairly low magnification, where you can see the little cilia, the hair-like protrusions at the top right of the slide. And underneath there is an electron micrograph, an exaggerated view of these cilia. And these cilia beat in unison, and they act like an escalator, moving the debris from the lungs. Often when you swallow, you're swallowing up the debris brought up by the lungs, and it then goes into the gastrointestinal tract, where it is eliminated eventually. So going on to the skin with a little bit more detail, the skin, the external layer of the skin is the epidermis. This consists of a layer of keratinized dead cells, keratin being a protein, which adds a sort of hard layer to it. The outer layer is the epidermis, and under that there is the living dermis. And many of you know if you cut yourself, you can cause pain. This is the nerve cells underlying the dead outer layer. You can cause bleeding. All living layers need to be oxygenated. Blood brings oxygen. There is the sebum and the sweat. The sebum is the oily secretions that prevent your hair and skin from drying out. And when you wash with soap, what you're doing is you're creating an emulsion of these oily secretions, which contain bacteria and other unwanted material. And then this emulsion is washed away with water. So you can hinder the immune system by excessive washing, for example. But the sebum, the oily secretions, the fatty acids, these have antimicrobial effects. They kill bacteria and they entrap bacteria, and these bacteria can then be washed away. But we have a normal skin microflora, which actually helps protect us. This layer of bacteria prevents the incoming bacteria from establishing, and often the incoming bacteria will contain pathogenic members, which under normal circumstances cannot establish on the skin. And some of the skin diseases, such as psoriasis, interfere with the normal functioning of the skin and can cause or can predispose to infections. The mucosa, these are the membranes that are covered in mucus. the lung, the gastrointestinal tract, the mouth, the vagina in women. And the mucus acts to protect. In the lungs, the respiratory organ, we have the cilia which protect. In the gut, shown in 2 slides on the right, we have villi, which are projections of epithelium underlaid by lamina propria, which contains the immune cells. We have peristalsis in the gut, which can suppress colonization. This is the process that normally expels the unwanted material out of the end of the gastrointestinal tract, but it can also act to get rid of large parasites and to some extent with smaller bacteria. Mucus in the intestines is produced by goblet cells. In the stomach, we have similar cells, mucus neck cells in the gastric glands, which also produce mucus. And again, the intestine, and to a much lesser extent the stomach, do contain their own microflora, which competes with the incoming pathogen. Now, many of you will know that ruminants, such as cattle and sheep, the area before the stomach, the true stomach, the epimazum, has become specialized into 3 pre-stomachs, including the rumen. And the rumen itself is packed with bacteria and protozoa, which play a major role in pre-digesting food. But they're not discriminatory. They will pre-digest the elements like cellulose that the mammalian digestive system cannot digest, but they'll also digest the other food as well. So to a large extent, although we talk about cattle and sheep eating grass, what's happening is the bacteria in the rumen and the protozoa in the rumen are eating the grass, and what the sheep and cattle are living off are the bacteria and the bacterial fragments that then pass to the stomach. And this has consequences, for example. So in mammals, we're adapted to living off sugars. So the brain uses sugar as food. Sheep and cattle, there's not a lot of sugar because it's pre-digestible bacteria. And instead, the main energy source there is small chain fatty acids like butyrate, propionic acid. And they're the main food sources for the brain. So we've had a digression, but the main point of that is there is a microflora, a microbiome in the gut, and it competes with pathogen. And one of the modern areas of immune research is looking to see how the microflora influences the immune response, both in the stomach, in the intestinal tract itself, but also perhaps in other parts of the body. And the short answer is it can have a major effect on the immune system. So this slide here, and most of the slides have been taken from the first edition of the textbook, but they also occur in the second edition. These are the innate defenses of the mucosal surface. So reading from the left, we have the endogenous microflora, usually lying in the mucus above the intestinal tract. We have the villi, which produce secretions. We have a dome, which is a specialized area of immune function, which has resident leukocytes, which sense the organisms in the intestinal tract and respond appropriately if they contain pathogens. And there is also peristalsis. But underlying it all is the epithelial barrier, which I hope you now realize is not simply a layer of cells, but is a layer of cells with lots of other things helping to maintain the integrity of the body. So the epithelial barrier is the first barrier that bacteria need to overcome. And it's pretty effective. And you'll see that, for example, if you cut yourself, often you will get a minor infection. It will become inflamed. And this indicates the effectiveness of the epithelial barrier. When it's broken, we get small infections, which can, in some circumstances, become serious. So that's the epithelial barrier. That's the first of the 4 barriers which underlie the effectiveness of the innate immune system. Now, normally at this stage, if I'm lecturing to you in person, I will stop, give you a chance to reflect on what I've told you about the epithelial barrier and ask if there's any questions. Now, we can't actually do that at the moment, so I'll proceed without further ado, onto the physiological barriers. And these are the second major component of the innate immune system. So we have temperature is a physiological barrier. Often, if you get an infection, your temperature will rise. We have what we call fever. And many people think that fever is induced by the body to try and limit the effectiveness of bacteria dividing. So it's a protective mechanism. Now, it can get out of hand. Temperature can kill. It's uncontrolled. But generally, the increase in temperature following infection is induced by the body as a protective mechanism to help fight infection. I've mentioned the low pH, the acidity of the stomach. Typically, the stomach, the pH is about 3 to 5. And this low pH acts to kill many of the invading or potentially invading bacteria. Now, some nematodes, and we'll talk about this later in the course, actually raise the pH of the stomach. They do this indirectly. The body, as part of the repair process, to repair the damage caused by nematodes, stops producing acid. This makes sense. If you have holes in your stomach, you don't want to be producing acid, which would then go in and damage your own cells. So the pH will rise following infection, and this can predispose to other infections. So the low pH is important. But there's a whole host of chemical mediators, chemicals that play a role in resisting infection. We don't have time to talk about all of these. It's an active area of research. People are actively discovering new chemical mediators. And the practical, the bioinformatics assignment that you are given and has been posted along with this lecture is about looking at one of these chemical mediators that plays a role in resistance to infection called galectin. Now, galectin is a family of molecules, but you'll need to do the assignment, the bioinformatics assignment, to find out more about them and their interaction with similar chemicals produced by nematodes. So dealing with the chemical mediators in a little bit more detail. Perhaps the classic one is lysozyme, which was discovered by Alexander Fleming, who was a Scotsman working in London, who also discovered penicillin. Lysozyme is in secretions, it's in tears, and it plays a role in digesting the membrane of certain types of bacteria. It's an important part of the innate immune system. The innate immune system also produces a whole series of antimicrobial peptides, of which the best known are the alpha defensins and the cathelicidins. They're able to, as the name suggests, interfere with the division of bacteria. If bacteria cannot divide, they're essentially dead. Cytokines, immune hormones. There are the interferons that are produced following infection with viruses. Initially interferon alpha, interferon beta. They play a key role in triggering a series of responses to viral infection inside the cell. Interferon gamma is another important molecule. This is produced by immune T cells, amongst other cells, and this plays a role in shaping the adaptive immune system and its response to infection. There are a series of interleukins. Over 33 have now been identified. Two of the most important are interleukins 1 and 6, which are produced as part of the innate immune system and play a key role in triggering the innate immune response to infection, but also in preparing the adaptive immune response to respond. And tumor necrosis factor, another cytokine that's produced, originally named for its role in causing death of tumor cells, hence the name, tumor necrosis factor, but it plays a key role in lots of other processes as well. Acute phase proteins, following infection, a series of proteins will increase enormously, or some of them occasionally will decrease the concentration, but the rapid increase in concentration of certain proteins, such as C-reactive protein and haptoglobin, can be used to signal a response to infection. So they have a role in diagnostics. But within the body, we think these proteins are increasing in concentration following infection as part of the innate immune response. These proteins can play a role. So C-reactive protein can help phagocytosis because macrophages and other cells contain receptors for C-reactive protein. So C-reactive protein can bind bacteria, and the body can bind C-reactive protein, digest it, and then kill the ingested bacteria. Haptoglobin is another important molecule. One of the main defenses against infection is to sequester iron, which bacteria need. Haptoglobin sequesters iron, so bacteria lacking the iron they need to divide will die. And there are lots of other acute phase proteins as well. But perhaps the most important and the most interesting of the chemical mediators is complement. This is a group of serum proteins. There's a cascade of pathways. And what they do at the end of the pathway is they punch holes in foreign cells, which allow, because the concentration of protein is much higher within a cell than outside, there is an influx of fluids attracted by the need to maintain osmotic balance. These inrushing fluids cause the cell to burst and die. But complement has other roles as well. It can help with phagocytosis. And in fact, complement is so important, we'll devote a special lecture to the functioning of complement. And this will be lecture 3. Complement works particularly well with antibodies in the classical pathway. And it's probably true to say without a functioning complement system, we would be in serious trouble. So we have the epithelial barrier, we have the physiological barrier, and the third barrier is the phagocytic barrier. And as the name suggests, this contains the phagocytes. And the picture on the right shows a macrophage sending out a projection, and you can just see at the very bottom right of the picture a bacterial cell which is being sought by the phagocyte. Now, on a microscopic level, that looks pretty scary, and so it should be. The bacteria has very little chance of evading the ingestion by the phagocyte. It's a primitive defense system. It was developed fairly early on, well before the development of mammals. But it's an effective one. Cellular ingestion of pathogens and foreign particles. And the phagocytes can, in a sense, be thought of as vacuum cleaners. Now, possibly their original role was to tidy up debris within the body, recycle used proteins, but they've evolved to play a role in defense against infection. There are a whole series of phagocytes. There are monocytes in the blood, which leave the blood, go into the tissues, and differentiate into a whole series of other cells. Now, macrophages and dendritic cells have quite different functions. Macrophages are strictly immune defense. Dendritic cells, their major role is to activate the adaptive immune system, but they both ingest foreign material. And in different parts of the body, they have a distinct appearance and they have slightly different names. So in the liver, for example, the macrophages are known as Kupffer cells. In the skin, the dendritic cells are known as Langerhans cells. And we'll show you a picture of these later. In the brain, they're microglial cells. In the lymph, the blood, the lung, and other tissues, they're known as macrophages. And in the blood, we also have plasmacytoid dendritic cells, which play a role in phagocytosis. They're important in immunity. They process pathogens and present fragments of pathogen molecules to the immune system and tell the immune system what to respond to. They're assisted by antibody and by complement. And phagocytes form the antigen-presenting cells. So the classical antigen-presenting cells, dendritic cells, macrophages, and B cells, which also produce antibody. And this slide here is an example of phagocytosis. So the initial stage here in the macrophage, there are microbes, which the macrophage ingests, so that it gets close to the surface. And macrophages are often attracted to microbes by the chemicals that microbes produce, or they can be attracted by chemicals that the immune cells are producing, which are essentially saying, there is an infection here, come and fight it. So they put out pseudopodia. Now, pseudopodia is a fancy name derived from the Greek, meaning false feet. What happens is projections of the cell go out, they surround the microbes, shown in the second picture, they fuse together, and then microbes are essentially trapped within a vesicle. This vacuole is then fused, typically with lysosome. So you get a phagolysosome containing enzymes which contains enzymes and other toxic compounds which digest and destroy the microbes. And then this vacuole then fuses with the external membrane, and the microbial debris is released by exocytosis. So it's a pretty effective system. And macrophages are known as professional phagocytic cells because they can ingest and destroy bacteria again and again and again and again. They're very effective. And they need to be. Bacteria can divide rapidly if not controlled. Now, there are additional cells of the innate system. We've talked about dendritic cells, macrophages, the roles they play in phagocytosis. But we also have neutrophils, which are also very effective phagocytic cells. Eosinophils, they're not phagocytic. They play a major role in defense against larger parasites, particularly nematodes. And many people think the main role of eosinophils is nematode. But eosinophils are probably the great neglected cell of the immune system. Because in the West, we seldom have large parasitic infections anymore. They've been controlled, particularly by good plumbing and sewage treatment, to a lesser extent, but still effectively by modern drugs. Because they're not part of Western medicine, they tend to be ignored. But we still find large numbers of individuals who are heavily infected with parasites. And it's only within living memory, for example, that rich countries like Japan and South Korea have become free of parasitic infection. So eosinophils are important. Mast cells are important. They're the key role in late onset inflammation. Natural killer cells, these cells play a key role in resistance to viral infection. And we'll talk about these in more detail later on in the course when we talk about resistance to infection. But we also have intraepithelial cells. These play a key role in various processes. So neutrophils, we have a diagram of a neutrophil here, plays a key role in phagocytosis. But unlike macrophages, neutrophils are not thought of as professional phagocytic cells. They ingest a small number of bacteria and then essentially they die. Neutrophils can degranulate, and again, they will die following degranulation, but they release a whole series of granules which contain substances that are toxic for bacteria. So they contain antimicrobial peptides. With phagocytic cells, they will release reactive oxygen species, which can kill, reactive nitrogen species, which can also kill microbes, and they contain hydrolytic enzymes. And a new mechanism, which is used both by neutrophils and eosinophils, is the release of extracellular traps. Usually the cell dies, but not necessarily. It then essentially, it coughs up its guts, it spills out DNA, lots of proteins, and this forms a sticky web that can then trap bacteria, prevent them moving and infecting healthy cells. And extracellular traps are an area of intense interest in modern immunological research, particularly with neutrophils, but also with eosinophils. Okay, so we've dealt with three barriers of the innate immune system. We've dealt with the epithelial barrier, we've dealt with the physiological barrier, we've dealt with the phagocytic barrier, and now we're going to talk about the fourth and final barrier, which is the inflammatory barrier. So you get a breach of the epithelium, say, for example, a cut shown here in the leg of a horse. You get tissue damage by abrasions, cuts, and wounds. And classically, you will get inflammation. Now, all of you have probably observed this if you've cut your finger or you've got a splinter which becomes inflamed. And the classic signs of inflammation are fourfold. They're hot, red, sore, and swollen. Some people talk about a fifth sign, which is a loss of function. But in practice, this is a consequence of being sore and swollen and is often only a partial loss of function and is not a necessary attribute of inflammation. So the four classic signs: hot, red, sore, and swollen. Now, inflammation, I've shown here a slide on the right, is often caused by an influx of cells. This inflamed canine skin here. You get tissue damage, recruits mast cells, macrophages, dendritic cells releasing chemicals. These chemicals cause vasodilation, which is a swelling of the blood vessels. This increases the capillary permeability, releases fluid, hence the swollen aspect, but it also allows phagocytes to enter the tissue with the aim of digesting any bacteria that have come in. The chemicals also mediate chemotaxis, which is the movement of cells along a chemical gradient. So there's an attraction of phagocytes to the infection site. The classic phagocytes are macrophages and neutrophils. And there's extravasation, where the phagocytes migrate via the capillaries to the damaged tissue. They then leave the tissue, often through the increase in capillary permeability, and then they can attack any bacterial infection at the site. So inflammation, although it can be useful, can be painful, is very, very useful, and it plays a key role in fighting infection. Inflammation only becomes a problem when it persists, so chronic inflammation. So there are two distinct phases. There is an acute inflammation. What we've got here on the right is an alpaca lung taken from the textbook, and we see a mass of cells around a mycobacterial, site of mycobacterial infection. Often these will form granulomas, so they're walled off. So the two acute phases of inflammation. The acute phase takes place within minutes. And again, we have these four signs: pain, swelling, heat, redness. Occasionally there is loss of function. And acute inflammation is mediated by neutrophils. Neutrophils leave the blood and do their job. Chronic inflammation takes place over 24 to 48 hours and is mainly driven by monocytes from the blood maturing into macrophages, which become activated. And these long-lived phagocytes form the second line of defense. And some of the inflammatory diseases, such as inflammatory bowel disease, which is a particularly painful problem of the gut, are caused by macrophage-mediated rather than neutrophil-mediated inflammation, although neutrophils do play an occasional role in inflammatory diseases. So this slide here expands upon inflammation. So number one, bacteria and other pathogens entering the wound on the right. Then we have platelets from the blood, releasing blood clotting proteins, which prevent the bleeding. This is why hemophiliacs are in so much trouble, even from minor cuts, because they're unable to stop bleeding. Mast cells, which are usually resident in the tissues underlying the epithelial barrier, these secrete factors that mediate vasodilation and vascular constriction. So in some areas, the blood vessels dilate to let the phagocytes out. In some areas, blood vessels contract to prevent bleeding. Delivery of blood, plasma, and cells to the injured area increases. So we get the fluid causing the swelling. Neutrophils recruited from blood secrete factors that kill and degrade the pathogens. Neutrophils and macrophages also remove pathogens by phagocytosis. Macrophages also release factors, hormones called cytokines, as do mast cells, that attract immune system cells to the site and restart the process of tissue repair. And the inflammatory response continues until the foreign material is eliminated and the wound is repaired. So inflammation is a pretty well coordinated and effective process to deal with infection at the site of a wound or an area where infection takes place. It doesn't necessarily have to be a wound as such. Some insects can inject substances through their proboscis, essentially their long nose. So there is a military analogy here, which is a bit childish, but still contains the right issue. And again, this comes from the textbook. So we have the invading pathogens in blue. They break through the barrier, shown here by a group of trees, although in practice in the army, I suspect to be much more effective than a forest. You might have trenches, barbed wire, for example, mines possibly in some areas of the world. But anyway, we have the innate immune defense comes in, tries to fight the invading pathogens, and what it does is it sends a messenger to the organized lymphoid tissue, such as the lymph nodes, that the adaptive immune system needs to get activated, and the adaptive immune system then supplies the reinforcement to the innate immune defense. So like all analogies, you can take it too far, but basically it conveys the thing. We have a patrol, a border force, which is the first line of defense, but is able to call on substantial reinforcements if needed to help fight the infection. And again, we come back to this diagram. So the antigen crosses the body. It's met with by the innate immune system, complement, antimicrobial peptides, enzymes, neutrophils, macrophages, natural killer cells. It's also picked up by dendritic cells, which migrate to the secondary lymphoid tissue, particularly the lymph nodes, where we have activation of T cells, which in turn activate B cells, as well as other T cells. These activated cells move into the blood, and from the blood, they go to the site of infection and fight the incoming organisms. So hopefully this diagram makes a lot more sense to you than it did when we started this lecture 40, 50 minutes ago. This slide here, and we're coming to the end now, is talking about the evolution of the immune system. We have phagocytes present pretty much everywhere, protozoa, sponges, annelids, arthropods in the invertebrates, and also in the vertebrates themselves. Natural killer cells are found in vertebrates, also in annelids, but not in most invertebrates. Antibody as part of the adaptive immune system, and the adaptive immune system only exists in the higher vertebrates. The MHC is a key set of molecules that, in a sense, act as the conductor of the immunological orchestra. These play a critical role. And the MHC is found in the higher vertebrates, some of the teleosts, some of the amphibians, but definitely in reptiles, birds, and mammals. T and B cells are found. They are, in a sense, they define the adaptive immune system. But lymph nodes, the organized immunological tissue, are only found in some birds and in mammals. So the immune system, the innate immune system in its simplest, phagocytes, exists throughout the animal kingdom, but the adaptive immune system is really a product of higher vertebrates. But you can see in an evolutionary sense, we've been groping towards an effective immune system for some time. Primitive defense systems of soluble molecules, antibiotics, penicillin, for example, was discovered in Penicillium, and it uses it to fight bacteria. Phagocytes, which is a yeast. Phagocytes themselves are found in most organisms. They engulf molecules and whole cells and are critical for host defense. So in review, I've introduced you to the innate immune system, which is primitive, non-specific, fast-acting, memoryless, and it might help you to think of the innate immune system consisting of 4 barriers. There is an epithelial barrier consisting of the skin, but also the mucous membranes underlying the gastrointestinal tract, the lung, other areas of the body that can, in a topological mathematical sense, be considered as external to the body. Physiological barriers, fever, for example, pH of the stomach, the phagocytes, in particular the professional phagocytes, the macrophages, but neutrophils also play a critical role in phagocytosis. And if your neutrophils, for example, are prevented from getting to the site of infection because you have a genetic problem, then you have a serious problem and you're unlikely to survive for very long. So one of the major diseases of cattle until quite recently, dairy cattle, was a genetic defect that prevented neutrophils leaving the blood and fighting the source of infection. And most of these calves were dead by 6 months of age. All of them were dead by a year of age. So phagocytes are critical and crucial. And then there's the inflammatory barrier. And we talked about how inflammation works. Classic signs of inflammation, hot, red, sore, and swollen, are basically an indication that the immune system is doing its job and fighting infection. This lecture is based quite closely on chapter 1. I'm sorry, this shouldn't be labeled lecture 2. This is lecture 1. This is part of the change in the way we've done the course. For lecture 2, you should read chapter 2, which is about antibodies and antigens. We're going to talk about adaptive immunity, and we're going to introduce antigens and antibodies. So I hope you've enjoyed this lecture. I hope you have been informed as well as entertained. I hope you will come and listen to the second lecture. And if you found this lecture complicated, and I'm aware we're introducing a lot of material, it will help you to look over the slides beforehand, and I will post these soon, well before lecture 2 is due to be delivered on Tuesday. But it might also help you to read chapter 2 in the book. Now, you can buy a copy of the book, and it is an excellent book, but the library also has copies that you can access online. But however you access it, I strongly recommend that you read the book. Thank you very much. And goodbye.